Okay, so this is the last little tiny bit of chapter 15 that we weren't able to get to in lecture. Okay, so the first technique that I want to talk about is pulse field gel electrophoresis. And it is very similar in many ways to genetic fingerprinting. So probably everybody who's ever watched CSI or any of these shows knows that what they do is they can take a person's DNA and they can chop it up in certain ways and run it through a gel, and you get this fingerprint based on where the actual bands are on the gel. Um, so this technique, this PFGE technique, involves the separation of DNA fragments that, generally speaking, are too large for conventional gel electrophoresis methods. So part of the problem when you have large fragments of DNA is it's just physically difficult to get it through the gel. And so here they're able to separate it using alternating voltages. So they do charge up top, charge at the bottom. And so very quickly, what ends up happening is that these fragments end up separating over time because they're being pulled in both directions. So ones that are a little more positively charged might move a little faster towards the negative pulse, and the ones that have more negative charge might move a little closer to the positive. And so the nice thing is it allows even similarly sized DNA fragments to separate on the gel into different bands. The DNA is subjected to restriction enzymes, and we talked about that a little bit earlier. These are those enzymes that recognize particular places in the DNA and snip, leaving those sticky ends. Okay, so by using that, basically it's looking for mutations in particular kinds of places. And anytime you have a mutation there, you're going to end up with a different sized band because the restriction enzyme is not necessarily going to be able to recognize its six-letter site. And so the band will not be snipped, or in the case of a mutation that causes it to be that restriction fragment, the one that it recognizes, it will cause it to be snipped. And so you get those different bands based on where those mutations are. This technique is widely used to look for outbreaks of foodborne illnesses or other kinds of infections, things that can be found using this kind of genetic technique. Okay, so there is something called PulseNet, which is established by the CDC that lets them track disease outbreaks, especially ones caused by foodborne pathogens. So basically, people from all over the country, places where these outbreaks are occurring, can use this technique, and then they can essentially send the results to the CDC. And the CDC is then able to collect information from all of these different places and get that microbial fingerprint and know if it's being caused by the same organism. And if it is, then they're able to start tracking it back to the original source. So the recent E. coli outbreak from ground beef was able to be tracked back to that particular manufacturer very quickly because they were able to fingerprint those microbes and realize that the ones in one state were the same as the ones in another state, and then they were able to track back where everybody bought their ground beef and stop it at the source. Okay. So here you're talking about identification of outbreaks taking hours instead of days or weeks, where they're having to grow organisms and so on. Now, that said, the CDC is in the process of converting to a whole genome sequencing as opposed to this PFGE, because the whole genome sequencing gives you additional information over and above just, these particular genes that they're looking for. Okay, so here's a picture of PFGE. So the bacteria are basically put into a buffer. They then actually add some agarose to the tube, and that basically is making it like Jello. Okay, so it basically fixes the bacterial sample. That it's referred to as a plug once it's mixed with that gel. So the gel is then subjected to a solution that pops open the bacteria. It's referred to as lysis. So they put a lysis buffer, or they apply a lysis buffer to the plug. So then all of the cells are broken apart, and they then have access to just the DNA. And then once they have access to the DNA, they apply the restriction enzymes, which start to cleave the DNA within the plug. Then what they do is they put it into the gel that they're running on. They're doing the pulses to be able to separate it. And they are able to get the DNA to come out of the plug the way it does. It runs through any other part of the gel, and then be separated on the gel using those the pulses that we talked about. Okay, so that is the end of chapter or of section 15.5. So here's the question. Fluorescent in situ hybridization, which is referred to as FISH, requires a pure culture of organisms obtained from the patient sample, true or false? And the answer is false. So one of the benefits of using this particular kind of technique is that we have those probes that have that fluorescence label. And the probes are very specific to only one particular gene on one particular bacterium. And so you can very easily identify the presence of a particular organism even in a mixed culture, like you would often get with a patient sample. Okay, so just a little bit more. We have section 15.6, and here we're going to talk about something called lab on a chip, and we're also going to talk about a technique called mass spectrometry, and also imaging, which is things like CAT scans and PET scans and all that sort of stuff, using those as diagnostic tools. Okay, so here's a picture of what's called a lab on a chip. And basically what they're able to do is they're able to have a number of genetic tests miniaturized and put on one chip. And essentially what that does is it keeps labs, particularly labs in developing countries who probably don't have access to a zillion different reagents and refrigeration even in some cases. So this allows those genetic tests to be kind of a one-stop shop. So if you have this lab on a chip, you're able to apply whatever it is that it's looking for. So DNA or RNA and PCR products can be placed on this chip and then run through this whole battery of genetic tests in one single place. And what this does is it allows them to be able to capture that information from the sample without having to have all of those different reagents and so on. And then they get access to their data once the chip has been analyzed by a place that does have the ability to do that, but everything is captured at the original site. Okay, so mass spectrometry, spectrometry, uh, not, I hate that word. It's basically a protein fingerprinting technique. So basically what you need is a pure culture, some kind of isolate from a patient specimen. Okay, and then what you do is you take that sample and you add it to a metal plate and you zap it with a laser. And that ionizes the sample and kind of breaks it apart a little bit. And it allows that sample to be looked at using mass spectrometry. And what that does is it looks at a mass to charge ratio. So what you find is that different ions, different elements in a protein have different sort of wobbling ratios between mass to charge, and it's kind of how it wobbles. And so the mass spec is able to look at all of these proteins and be able to identify what the things are. And also, it can often detect antibiotic susceptibility while it's doing that from the presence or absence of particular protein products. So here's an example of some patient samples. So the one up top is non-infected. And then the one in the middle is slightly infected. And so you can see that some of the bumps in the readings have changed. And then ones that are heavily infected, you see that the height of the bumps in certain places are considerably higher. And it's that the height of the bump and the place that the bump occurs that's used to read these particular charts. Okay, and then the last thing we're going to talk about is the use of imaging. So X-rays have been around for quite a while, at least a century plus a little bit. I think it was the late 1800s when they came into use. And they have been used in the diagnosis of tuberculosis because obviously you had to be able to look inside the lungs to see if the lesions were there. Nowadays, we have a number of different imaging techniques, including MRI, CAT scans, PET scans, all of that sort of stuff. The nice thing about it is that it's able to look inside of tissues without having to go in surgically and biopsy those. And so if what you're looking for is an abscess, you won't necessarily have to cut somebody open to look to see if it's there. You can use these imaging techniques and not only know that it's there, but know exactly where it is and be able to come up with a strategy of how you can get in there without damaging tissue in order to find it and get rid of whatever the problem is. Okay, um, so there are also some additional new diagnostic types of strategies. And in one case, they use patient blood and they look for the presence of 7 specific genes that host cells, in other words, our cells, express in response to a bacterial infection, but not a viral infection. And basically, if the genes are active, then they know that we have a bacterial infection and that they need to be prescribing some sort of antibiotic. However, if those genes are not active, then you pretty much know that you have a virus and an antibiotic is going to be completely ineffective. And so it's really useful where so many prescriptions for antibiotics are prescribed for viral infections. And so, you're exposing somebody to antibiotics with all of the downside as far as their microbiome is concerned, for absolutely no benefit. It's not going to affect the viral infection. So there's also some research going on in Boston where they're developing some blood tests similar in nature, but here they're looking for antibodies to more than 1,000 strains of different viruses, representing 206 species. So these are the ones that most people are getting sick with. And they're able to identify positively that there is a virus involved as opposed to a bacteria. So then you have to look for antiviral strategies and that you definitely do not want to prescribe an antibiotic. Okay, so here's our concept check. Which of the following technologies detects microbes via their protein fingerprints? So the answer is B, mass spectrometry. So nucleic acid sequencing, of course, that's nucleic acid, not protein. Microarrays, again, are looking at nucleic acids. Okay, so here you're doing basically a large series of genetic tests looking for particular mutations. So that again is nucleic acid. Imaging is looking at the whole body, so it's not looking at protein or nucleic acid. It is taking pictures of large sections of the body, looking for evidence of some kind of an infection, like an abscess, or it's also used for things obviously like tumors and things like that. And immunofluorescence is a lab test where you're looking to see if you have a particular antigen inside, for example, a blood sample, using antibodies that are labeled with some kind of a fluorescent tag. And so if the antibodies stick, that means you have the antigen present, and if they don't stick, they get washed away, then there's no fluorescence and therefore you don't have it. But again, it's looking for antigen, not looking for a protein fingerprint.